morning everyone. We'll begin with the next chapter or the next part of the chapter, that is the cell, the unit of life. In the previous classes we have seen the endomembrane systems. Now we will go with the next organelles, that is the mitochondria. Hope you have heard about the mitochondria. They are called as the powerhouse of the cell. Why it is called as a powerhouse of the cell is a question. Or what is the importance of that mitochondria? How you are getting the energy in your body to do all the metabolic activities. The cells are getting the energy to do the activities. In turn, you are getting the energy to do the activities. So what is that structure that is helping in to provide energy is the mitochondria. You can just look into the chart, the figure, the structure of a mitochondria, or you can just refer the textbook and see at the textbook for the structure of a mitochondria. The mitochondria is one of the important organelle which is present in both the plant cells and the animal cells. Cells require energy to do the work. So that energy is provided by the organelle, that is the mitochondria. It was first observed by Rudolf Collier in the muscle cells. He observed the mitochondria in the muscle cells. The scientist who described or discovered the mitochondria was Rudolf Collier, but the term was coined by Carl Bender, the scientist who named it as mitochondria. It is derived again from the Greek word, that is mito, thread-like, chondrion, granules. They are thread-like granules. The shape, they also differ in shape, but usually they are rod shaped. They are rod shaped or sausage shaped. The scientist, when he observed these are present in the cytoplasm of the plant cells and the animal cells. They are scattered, they are distributed, or they are suspended in the plant cells and the animal cells. So here, if you go with the structure, there are the mitochondria, which appears to be sausage shape with an outer membrane and the inner membrane. This is the outer membrane, outer layer, and this is the inner membrane. Or the expanded figure is being drawn on the board. So the outer membrane of the mitochondria is around 60 to 75 angstrom. There is an outer membrane. In simple way, let me go with another. Just look into the diagram. It is having an outer membrane, inner membrane. Sausage shape with outer membrane. This outer membrane is permeable. It is permeable around 60 to 75 angstrom thick. Outer membrane is permeable around 60 to 75 angstrom thick, and it is having a inner membrane. The inner membrane, which is around 50 to 70 angstroms. So 50 to 70 angstroms. And the space between the outer and inner membrane, this space, this is the outer membrane, this is the inner membrane, the space is known as mitochondrial Membrane, it is a double membrane organelles where the outer membrane is permeable with 60 to 75 angstrom. The inner membrane, this is the inner membrane with 50 to 70 angstrom unit. Then the space between the outer and the inner membrane is known as perimitochondrial space, which is about 80 angstroms. If you come to the number of the mitochondria, as it is covered in the muscle cells, more other cells were been observed and to find out the number of the mitochondria. So in the kidney cells, you can find around, in the kidney cells, there are around 300 to 400 mitochondria, while in the human sperm cells itself have 25 mitochondria. The liver cells have 1000 to 1600 mitochondria, while the flight muscle cell has around 50,000 mitochondria. So the mitochondria varies on the metabolic activities or varies on the positions of the cells. In the kidney cells, there are around 300 to 400 mitochondria. In the human sperm cells itself, a single human sperm cells have 25 mitochondria. In the liver cells, there are 1000 to 1600 mitochondria. In the flight muscle cells, especially in the birds, that are the locomotory organs, the wings, the locomotory organs of the birds have around 50,000. So as with the structures, so we know the number, how many number of mitochondria. So here it is having an outer membrane that is permeable. The inner membrane is selectively permeable. Already in the previous class I said you what is permeable and selectively permeable, impermeable. Once again, what is permeable? This is a layer which is allowing the substances to pass through. All the layers, all the membrane of the organelles have similar structure of that of the plasma membrane. So here it is permeable, while the inner membrane is selectively permeable. That is, it does not allow most of the substances to pass through it, only selected materials to pass through it. Then the most important feature in the mitochondria is, you just can observe here, the inner wall has in the inner side some infoldings. There are inner infoldings inside the mitochondria. Now these inner infoldings, the plasma membrane is producing a finger-like projection internal to the body cavity, into the cavity. Those finger-like projections are called as the cristae. The plasma membrane has inner folded finger-like, or it increases the surface area where it is having inner folded projections. These inner folded projections are called as cristae, singularly as a crista. The inner membrane has inner foldings, have the inner foldings. These inner foldings are known as the cristae, singularly is the crista. Then this inside the cavity of the mitochondria, there is a fluid proteinaceous substance. This fluid proteinaceous substance is known as mitochondrial inside the mitochondria, where it contains the proteinaceous substance. So here the components. Mitochondria is known as the powerhouse of the cell. So it should have those substances that help it to produce the energy. So all those chemical substances or all the substances, molecular substances are present in the mitochondrial matrix. So it is a proteinaceous submatrix or a fluid matrix where it is having the enzymes that are required for the process of aerobic respiration. It is containing the enzyme. The matrix is containing all the necessary enzymes for the process of aerobic respiration. What is aerobic respiration? There are two forms of respirations. Respiration that occurs in the presence of oxygen and the respiration which occurs in the absence of oxygen. So here the mitochondria in the presence of oxygen undergoes the process of respiration. The respiration which occurs in the presence of oxygen is known as aerobic respiration. So aerobic respiration it is. So it contains, this matrix contains the enzymes that are responsible for the aerobic respirations. In this apart from that, the matrix has, or the mitochondria has its own DNA. It is an independent unit or an independent structure with its own DNA, which is a circular DNA. It has 70 S ribosomes. S, try to remember this word, yes. So the inside the matrix it contains the 70 S ribosomes along with the RNA and few amount of starch grains. A few amount of the starch grains. So in this matrix of the mitochondria, you will find a circular DNA. There is 70 S ribosomes and the RNA and all the required enzymes to carry out the process of respirations. So it is a respiratory unit where it undergoes the respiration process. So once again we will look with the structure. So mitochondria was discovered or observed by Rudolf Collier but was named by Carl Bender. He called them as mitochondria because they appear like thread-like granules. They are sausage in shapes. So here it has an outer membrane which is permeable, inner membrane which is selectively permeable, and the space between the outer and inner membrane is the perimitochondrial space. The inner membrane shows the finger-like projections known as the cristae or the crista, yes, which encloses a cavity. There is a cavity inside which is filled with the fluid, proteinaceous fluid. That proteinaceous fluid is called as the mitochondrial matrix. This mitochondrial matrix contains double Each of this crista has studded with, they are studded with knob-like structure. Let me expand this diagram. This is the outer membrane, the inner membrane, the cristia. Yes, this cristia have studded with knob-like particles. These knob-like particles were discovered by Rackers, so they are also called as Rackers particles or oxysomes. They are called as Racker particles or oxysomes. So if you just observe here, outer, inner layer, DNA, crista. Each of this crista have certain knob-like particles. The inner membrane has the cristia. The cristia are studded with knob-like particles. These knob-like particles are known as the Racker particles or the oxysomes. Further, these particles, when studied, showed the following parts. The oxysomes, each of the oxysomes showed the following parts. And the oxysomes were observed, described by Fernand's Moran, so they are also called as Moran particles. Moran particles. Each of the oxysome has a base, stalk, and the head, which are designated as F1, Moran particles. Fernand's particles, they are designated as the base as FO and the head as the F1. So here, during the process of respiration, with the help of the enzymes, the hydrogen ion are being produced on the outer surface. When they pass through the base and the head, it helps in the synthesis of the energy component, that is ATP. In the future classes, when we deal with respiration, you will be coming to know that how the synthesis of ATP occurs or how they are involved in the process of formation of energy or how is the energy released by the mitochondria. But just in a short, the protons when pass through these oxysomes, when they are passing through the oxysomes, they are helping in the formation of energy, that is ATP molecules. ATP molecule, adenosine triphosphate, which is considered to be the energy currency of the cell. So the mitochondria help in the formation of energy. That energy is ATP, so we call them as a powerhouse of the cell. So just go through once again the diagram. The outer membrane, inner membrane, perimitochondrial space. Inner membrane has a finger-like projections called as the cristae. The cristae are studded with knob-like structures called as the Racker particles, known as the oxysomes. These oxysomes were having the base, stalk and the head, mainly involved in the synthesis of ATP molecules. Coming to the functions of the mitochondria. What are the functions of the mitochondria? Mitochondria is known as the respiratory unit which undergo aerobic respiration through the process called as Krebs cycle and electron transport system. It is a respiratory unit which undergo aerobic respiration. This part represents the process of Krebs cycle. This Krebs cycle helps in the formation of the energy component. Another process simultaneously that is related to the respiration process is electron transport system. With the help of Krebs cycle and electron transport system, it is synthesizing the molecule ATP. So Krebs cycle and electron transport system you are going to study in the process of respiration chapter. Then it is producing, as a respiratory unit, it is producing the ATP molecule, adenosine triphosphate. As it produces the energy component ATP, the mitochondria are called as the powerhouse of they are known as the powerhouse of the cell, that component that produces or synthesis the energy. And this energy is used by the cells for conducting all the metabolic activities. So this is about the mitochondria, its structure and the functions. Next component is, or the next organelle are the plastids. Next organelles are the plastids. Let me give some example. Mango, lemon, chili, tomato, yes, capsicum, yes, fruits. The fruits, there are different varieties of fruits. Take even the vegetables also, like carrot, or you take the leaf itself. The plants have different colors. They represent in different forms. There are different varieties of flowers with different size, shapes and beautiful colors. How did that all color come from? Yes, the mango, initially it is represents with 3 color. Young, when it is raw, it is green in color, changes to yellow, then orange red. The lemons, raw, are green in color. When completely developed, forms yellow in color. The chilies, Chilies green in color change to form red in color. Tomatoes, capsicum, and the characteristic color of the carrot is the orange, and the leaf are green in color. So there are certain pigment organelles present in the plants. Those coloring pigments present in the plants and the euglenoids are the plastids. These plastids are the coloring pigments that are present in the plants and euglenoids. Plants and euglenoids. All the different varieties of the fruits, they have their own characteristics color. That color is due to the presence of the plastids. These plastids were first observed by scientist named Skimper. He observed, the first scientist to observe the plastids was Skimper. He called them as the coloring pigments. But mango outer cover is green, orange, yellow to orange, red. Lemon also green to red. Green to yellow. Chili green to red. Tomato also green to red. But when you take the seeds of these, the mango seeds is white in color. The lemon seeds are colorless. They don't have any particular color. So in the plastids, there are two forms of plastids. Those are colorless plastids and colored plastids. Colorless plastids and colorless plastids, they are the non-green part of the plants. Non-green parts of the plants, while the colored plastids are chloroplast, and the chromoplast. There are two plastids observed. One is the colorless plastids and the others are the colored plastids. Colorless plastids are the non-green parts of the plants, especially the seeds. Now, based on the substances they store, what are the reserved materials that they store, they are being classified. The colorless plastids are classified as Aeroblast, which store the proteins. For example, maize. Aeroblast, they are the colorless plastids which come under the section called as leucoplast. Colorless plastids are known as leucoplast, where are being classified based on the presence of the components they store as aeroblast, that is, which store in the form of proteins. Example is maize. Second is elioblast, which store oil and fats. Example, ground nuts. The groundnuts, yes, all those seeds or the components which store the oil and fats are elioblast. The groundnuts So in the plastids, these plastids usually develop from a set of meristematic cells. The meristematic cells group together and develop. They have a meristematic cells have set of cells known as protoplastids. Protoplastids which develop to form the plastids. So the plastids are the coloring pigments classified as colorless plastids and the colored plastids. Colorless plastids are the leucoplasts, which are being classified based on the components that they store as aleuroplast, elioplast or amyloplast. Then I said about the mango. The mango is initially green in color. That green in color, characteristic color of the mango is because of a green color component, that is chloroplast. So this chloroplast have a green color pigment known as chlorophyll, which provide the characteristic green color to the fruits, to the raw fruits and the leaves. While the carrots are characteristic color of the carrot is the orange in color. They have the pigment called as carotenoids. Carotenoids which provide a characteristic color to the fruits or the vegetables. So here can be interchange. There can be conversion from a leucoplast to chloroplast or chromoplast. For example, you take a mango. Mango is initially develop chlorophyll components which later on when ripened changes to form the carotenoids. So there can be here the chromoplast can develop from a leucoplast or the chloroplast can develop from a leucoplast. At particular period of time, there is change in the color of the fruits. There is change in the formation. Or raw fruits are appearing green in color, while the ripened ones appear in characteristic colors. So there can be change usually from a chloroplast to the chromoplast. Hope the chloroplast is clear to you all. Now next we will go with the structure of a chloroplast. So in the plastids, there are colored plastids and colorless plastids. Colorless plastids are the leucoplast. In the colored plastids, it's the chloroplast and the chromoplast. So just look to the structure of a chloroplast that is being given in the figure. So the chloroplasts are double membrane organelles. The chloroplasts are present in all the photosynthetic plants, that is the autotrophs, where it have a pigment called as chlorophyll which traps the solar energy and help in the synthesis of the food. So the plants have the unique characteristics green color because of the presence of the chloroplast. The chloroplast provide a green color to the leaves. So the chloroplast, the plastids that have a green color pigment are the chloroplast. The word chloroplast was coined by Skimmer. The term chloroplast was coined by the scientist Skimmer. It is a double membrane organelle with an outer membrane and an inner membrane. The number of chloroplasts are usually present in the mesophyll tissue of the cells. There can be around 20 to 200 chloroplasts in each of the cells. The cells, usually the plant cells present in the leaves. The meristematic cells of the leaves contain the structure called as the chloroplast, the green color component which is trapping the solar energy and help in the synthesis of food, in the formation of the food. So, we will go with the structure of a chloroplast. Look to the chart or to the diagram that is being given. It is a double membrane organelle. Again, it is having an outer membrane which is permeable. It is having an outer membrane that is permeable, which allows the substances to pass through it, while the inner membrane which is impermeable, The chloroplast in each of the plant cells are, if you take the leaf cells, there are two forms of leaf cells in the leaves, the palaceid cells and the mesophyll cells. In each of these cells, there can be around 20 to 30 chloroplasts in the meristematic tissues or in the mesophyll tissues. So here, the chloroplasts are usually discoid in shape. They are discoid in shape. In some of the chapters, you will find the shape. This figure also, or in the textbook, this form of figure. A simple diagram is being drawn to make it clear. So, it is having an outer membrane and the inner membrane. The outer and inner membrane, this space is known as periplastidial space, is known as periplastidial cell. Externally, it appears like a discoid structure present in the meristematic tissues, that is present in the mesophyll tissue of the plant cells. But when you take a section of it, and observe, there are two membranes with the periplastidial cells. In this, there are two main components. It encloses the two main components. This fluid proteinaceous component is known as stroma. So inside the chloroplast, there is a proteinaceous fluid-like substance. That fluid substance is known as the stroma. Along with that, you can just see that there are certain structures over here. You can just see that these structures. There can be, again, the number can vary from one cell to another cell. These structures are known as grana. These are grana. Singularly, they are called as grana. Inside the chloroplast, you can observe two main components, that is the stroma and the grana. The stroma is the proteinaceous component that is surrounding the grana. These are all the stroma component which surrounds the grana. This stroma is a site is the dark reaction. So there is a photochemical reaction. In that photochemical reaction, one reaction is known as a dark reaction that occurs in the stroma, while the grana are the site of light reaction, where the grana contain these all structures, this disc shaped structures which are arranged one above the other, these which are arranged one above the other like a pile of coins. Each of them are known as thylakoids. They are known as thylakoids, which are placed one above the other. This is of a single granum. This is a figure of a single granum where it is having the thylakoids. The number of thylakoids also differ. It can be from 2 to 20 or 10 to 30, differs from the activity of the cells. So these structures, the disc-shaped structures which are arranged one above the other are known as the thylakoids, where it is the place, or it is the site where the light reaction occurs. So the thylakoids are disc-shaped structures that are being placed one above the other like a pile of coins. Each of the thylakoids, if you take a single thylakoid, each of this thylakoid is also double membrane. It has two membrane, that is, this is the membrane and the lumen. Each of the thylakoids have a membrane and a lumen. In the membrane, they contains the photosynthetic pigments. The photosynthetic pigments, that is the chlorophylls and the carotenoids, are present in the membrane of the thylakoids. And the cavity inside the thylakoid is the lumen. So here I will give once again what are the stroma and the grana. The chloroplast shows two components, that is the stroma and the grana. The stroma is the fluid material that is present surrounding the grana or inside the chloroplast, where it is a site of dark reaction, the reaction which occurs in the absence of the light. While there are thylakoids. It has thylakoids. The thylakoids group together and are arranged like a pile of coins, which are known as grana. Singularly, it is called as a grana. Each of the thylakoids has a membrane with the lumen. This membrane have the photosynthetic pigments. So when the light falls on the thylakoid membrane with the help of that solar energy, these are a rough representation. This is a representation of the cycle where. Now here, if you just go through this chlorophyll organization, this is in detail about it, but in the chapter of photosynthesis you will be studying the complete structure of the chloroplast and how the process occurs, how they are absorbing the light energy, how they are helping in the formation of a component, or how they are synthesizing the food. But now with the reference, remember that the grana has the thylakoids. The thylakoids contain the photosynthetic pigments. These photosynthetic pigments help in in absorption of solar energy and are involved in the synthesis of the food. Then apart from that, these are the site of light reaction. Apart from that, the stroma, the fluid material. Here also the chloroplast has its own DNA. The chloroplast has its own DNA with starch granules and 70S type of ribosomes. Starch grains with 70S type of ribosomes. So the stroma, the fluid material, has the osmophilic droplets, the 70S ribosomes. Then just observe these, the connecting links between the granules, So one granule is connected to the another granule by a connection. There is a channel that connects one granule to the another. So these channels are known as fret channels or stroma lamella. So this is a granule, this is another granule. This granule, the two granules are interconnected by the channels. Those are known as the fret channels, which in turn help in the transportation of substances from one granule to another granule. So here you can see in this arrangement, these are the thylakoids which are arranged and the fret channels. So the granule are interconnected by the channels called as fret channels. When the leaves are green in color because of this component, that is the chloroplast. The chloroplast gives the characteristic green color to the plants. Similarly, the chromoplast. Chromoplast also have the similar components. For example, a lemon is changing from a green to yellow. So when a chloroplast dissolves itself and convert into chromoplast, that is, if chloroplast dissolves and convert to form carotenoids, the color of the fruit changes from green to yellow. Similarly with the mangoes or any of the fruits, where the raw will have the chloroplast. The chromoplast also have the similar structure, but the chromoplast will develop the characteristics carotenoid or the colored yellow or orange. Here the lemon or a capsicum or a chili, if you say that, the chloroplast, when the fruits loses the chloroplast, they become the chromoplast, where it loses the characteristic green in color to form the chromoplast. Chloroplast is converted into carotenoids. So when there is a change from chloroplast to chromoplast or a chloroplast into carotenoids, there is a characteristic change of the fruits from a raw green color to a mature or a ripened yellow, orange or orange-red in color. So these structures, so the plastids are the colorless and the colored plastids. Colored plastids are the chloroplast and the chromoplast. So in the next class we will be dealing with the other components of the cells. So thank you. Go through the diagram properly. Practice the diagrams when necessary. Thank you.